The topic is cell division, process of mitosis and meiosis. Firstly, cell divisions. The growth and development of every organism depend upon the precise replications of genetic material during each cell division. And the cell cycle, the sequences of events that encompass the period between the completion of one cell division until the end of the next cell division. Involves both divisions of the cell nucleus, names as the karyokinesis, and the divisions of the cytoplasm is the cytokinesis. And next, there are two types of the nuclear divisions. Firstly, meiosis and mitosis. Stomatic cells are formed by mitosis. Each cell division produces two daughter cells with the same number and the kind of chromosomes as the parent cell. And next, the formations of male and female gamete in animal cells or spores in the plant cells is by meiosis. And next, interphase. Interphase begins when cell division ends and continues until the beginning of the next round of the division. Is organized into three divisions. The nucleus and the cell increases in size and chromosomes are fully extended. The cell expends large amount of energy in the synthesis of RNA and the protein. And the next sections of the interphase is marked by the dramatic rise in the DNA synthesis. Synthesis of histones that are major cellular proteins bound to DNA. The cell is preparing for the beginning of the mitosis. The chromosomes are becoming longitudinally doubled with each chromosome consisting of two identical chromatids. Next is the mitosis. Mitosis is the next phase of the cell cycle. It is the process of coordinated chromosomes replication prior to cell divisions. The major functions of mitosis is to accurately and precisely replicate genetic information of chromosomes. So each daughter cells contains the same informations. And next, the process of mitosis is an ongoing event that can be segmented into several identifiable stages. During the mitotic phase, a unique complement of genes are activated. These stages are prophase, metaphase, anaphase, and telophase. Firstly, prophase. In prophase, dramatic changes begin to occur with the nucleus of the cell. Chromosomes become thicker and shorter. Two sister chromatids join near their middle and a structure called the centromere, we called it. Microtubules are slender rods of the protein responsible for pulling replicated chromosomes towards each half of the cell. The centromere split into two centrioles which moves to the poles of the cell. Next is the metaphase. During this period, chromosomes become aligned at a midpoint. These are easily identified as two longitudinally double sister chromatids. Chromatids are connected at their centromere to the spindle apparatus, which has formed between the two centrioles located at the poles of the cells. The plant chromosomes are similarly attached to the spindle microtubule fibers. Next, anaphase. In this short phase, sister chromatids begin to separate and migrate to the poles. Once the two chromatids separate, each is called as chromosomes. For example, onion has 16 diploid chromosomes and therefore 16 chromosomes move to each pole. During anaphase, there is a quantitative equal segregation of the diploid number of chromosomes into two developing nuclei at the poles of the anaphase cells. Next, telophase. The final mitotic phase of the cell cycle is recognized by the formations of two nuclei encompassing the daughter cells at the each cell. The mitotic apparatus disappears and chromosomes begins to lengthen as they wind. Next, cytokinesis. Cytokinesis, formations of the new cell membrane occur midway between the daughter nuclei. In onion root tip cells, there is a formation of the cell plate, dividing the original cell into two presumably equivalent daughter cells. Next, meiosis. Meiosis is a specialized type of the cell division sharing many features with mitosis. The main difference is that meiosis involves two successive nuclear divisions that produces four haploid cells. Each gamete or sex cell contains half the number of the chromosomes. Each gamete containing 23 chromosomes. Meiosis and meiosis 2 are the two rounds of the cell divisions of meiosis, and it has its own prophase, metaphase, anaphase, telophase. Firstly, meiosis 1, prophase 1. Firstly, the chromosome begins to shorten and thicken. They appear to aggregate together on one side of the nucleus. The first major difference between the mitosis and meiosis is that homologous pair of the chromosomes comes together or synapse. The point of crossing overs appears egg-shaped structure called the chiasma. And next, the metaphase 1. The synapsed homologous pair of the chromosomes arrive at the midpoint or equated between poles. The synapsed pair orient such that one member of each pair faces the opposite poles of the cell. There is no tendency for one member of the pair to face one member of the poles. Next, anaphase 1, the pair of homologous chromosomes is longitudinally double tetrad began to separate and migrate to the cell poles as contrasted to mitosis, entire chromosomes versus the cyste. And the next, telophase 1, the chromosomes arrives at the point of the cell at the beginning of this phase. The membrane forms and the nucleus begins to recognize cytokinesis. Physical cells divisions occurs during this phase. And next, meiotic divisions 2. In order to reduce the amount of DNA to half, a second meiotic division is necessary to separate the chromatids of the chromosomes in the two daughter cells formed in the meiosis 1. And next, prophase 2. This phase resembles mitotic prophase except the chromosomes do not dramatically shorten. The nucleus, the site of active or RNA synthesis, disappear. The nuclear membrane also disappear, and the mitotic apparatus, the spindle, begins to organize within the cells. And next is the metaphase 1, 2. The monoploid number of the chromosomes organized at the midpoint equated between the poles. Each chromosome is composed of the two sister chromatids.